So I am a scientist and also now a teacher, but I think that I have bounced around in and out of so many different areas of science that I really consider myself sort of a generalist, which works out really well for teaching, especially at the position that I'm doing now, because I kind of rotate around the different areas, and that has been a complete and total wild ride. So one of the books that I started actually last year and read over the summer is I Contain Multitudes. It's by Ed Yong, and he has an amazing YouTube channel where he talks about all things microbial. It's a phenomenal read if you feel like it. If you don't feel like settling into a read of this density, you can check out his YouTube channel. And it has been so enlightening, and he really does a phenomenal job of sort of giving an overview, but also making connections with so many different researchers and highlighting, but making understandable everything that they're doing. And so I come from a very scientific background, but I am also a mom. And I don't know that I started out to be some sort of militant nurser of my kids, but it really ended up being that way. I ended up nursing them quite long into their childhood, well beyond the year, and just had a really deep and really intense nursing relationship with both of them. And I always felt like breast milk was a better choice for me personally, and that there is so much that is beneficial about that based on the research that I understood at the time. But as I was reading this book, the I Contain Multitudes book, this really just blew my mind. So the one caveat to this before I start is, yes, fed is best. If you can't nurse your kid, if it's a problem, it's better that they're fed than not. However, it is absolutely unequivocally scientifically accurate that breast milk provides benefits that are absolutely unavailable in human-produced formulations. And there's some things that human babies can get from other types of animal milk. So there are some things, but that intimate relationship between the parent, between the mother or the nursing person and the infant, that is just irreplaceable. There's nothing that we are able to produce at the current time that is anything like it. So allow me to read this excerpt from this book. It's really amazing. So think back to the previous chapter in which I portrayed the immune system as a team of rangers carefully managing a national park. If microbes breach the park's fences, the mucus, the rangers push them back and fortify the barrier. They cull any species that becomes too dominant in the park, and they chuck out any pathogens that invade from the outside world. They keep equilibrium within the community and constantly defend this balance from threats both foreign and domestic. The rangers only get time off at the very start of our lives, when in microbiological terms, we are blank slates. To allow our first microbes to colonize our newborn bodies, a special class of immune cells suppress the rest of the body's defensive ensemble, which is why babies are vulnerable to infections for their first 6 months of life. It's not because their immune system is immature, as is commonly believed. It's because it is deliberately stifled to give microbes a free-for-all window during which they can establish themselves. But without the immune system's full selective powers, how can a mammalian baby ensure that it gets the right communities? It's mother's milk. Mother's milk is full of antibodies which control the microbial population of adults, and babies take up these antibodies during breastfeeding. When immunologist Charlotte Katzel engineered mutant mice that could not produce one of these antibodies in their milk, she found that their pups grew up with bizarre gut microbes. They were full of species that are typically found in people with inflammatory bowel disease, and many of these bacteria wormed their way throughout the gut walls to inflame lymph nodes lying underneath. As we saw earlier, many harmless bacteria are harmfulness only by virtue of where they are. Milk keeps them restrained, and it does much more than that. Milk is one of the most astounding ways in which mammals control their microbes. So this is part 2. I'm going to probably have to link these together later at some point or put them up as a series. At the University of California, Davis, there is a block of terracotta walled buildings overlooking a large vineyard and a garden bursting with summer vegetables. It resembles a Tuscan villa that has somehow been teleported into the western United States. It is, in fact, a research institute, and one whose residents are obsessed with the science of milk. They are led by a short bundle of nervous energy named Bruce German. If there was a world title in extolling the virtues of milk, German would surely hold it. I meet him in his office, shake his hand, and ask, "Why are you interested in milk?" Half an hour later, he is still monologuing his answer while bouncing on a gym ball and kneading a tattered shred of bubble wrap. Milk is the perfect source of nutrition, he says, a superfood that is actually worthy of the title. This isn't a common view. To date, the number of scientific publications about milk is tiny compared to the number devoted to other bodily fluids like blood, saliva, or even urine. The dairy industry has spent an unimaginable fortune on extracting more and more milk from cows, but very little on understanding just what this white liquid is or how it works. Medical funding agencies saw it as irrelevant. As German puts it, it doesn't have anything to do with the diseases of middle-aged white men. And nutritionists saw it as a simple cocktail of fats and sugars that could be easily duplicated and replaced by formulas. People said, "It's just a bag of chemicals," says German. It's anything but that. Milk is a mammalian innovation. Every mammal mother, whether platypus or pangolin, human or hippo, feeds her baby by literally dissolving her own body to make a white fluid that she secretes through her nipples. The ingredients of that fluid have been tweaked and perfected through 200 million years of evolution to provide all the nutrition that infants need. Those ingredients include complex sugars called oligosaccharides. Every mammal makes them, but human mothers, for some reason, churn out an exceptional variety. Scientists have identified over 200 milk human oligosaccharides, or HMOs, so far. They are the third biggest part of human milk, after lactose and fats, and they should be a rich source of growing energy for growing babies. But babies cannot digest them. When German first learned about HMOs, he was gobsmacked. Why would a mother spend so much energy manufacturing these complicated chemicals if they were indigestible and therefore useless to her child? Why hasn't natural selection put its foot down on such a wasteful practice? Here's a clue. These sugars pass through the stomach and the small intestine unharmed and land in the large intestine where most of our bacteria live. So what if they aren't food for babies at all? What if they are food for microbes? This idea dates back to the early 20th century when two very different groups of scientists made discoveries that, unbeknownst to them, were closely connected. In one camp, pediatricians found that microbes called bifidobacteria, or BIFs to their friends, were more common in the stools of breastfed infants than bottle-fed ones. They argued that human milk must contain some substance that nourishes these bacteria, something that later scientists would call the bifidus factor. Meanwhile, chemists discovered that human milk contains carbohydrates that cow milk does not, and were gradually whittling this enigmatic mixture down to its individual components, including several oligosaccharides. These parallel tracks met in 1954, thanks to a partnership between Richard Kuhn, chemist, Austrian and Nobel laureate, and Paul Grigory, pediatrician, Hungarian-born American breast milk advocate. Together, they confirmed that the mysterious bifidus factor and the milk oligosaccharides were one and the same, and that they nourished gut microbes. It often takes a partnership between different branches of science to understand the partnerships between different kingdoms of life. By the 1990s, scientists knew that there were more than 100 HMOs in milk, but had only characterized a few. No one knew what most of them looked like or which species of bacteria they fed. The common wisdom was that they nourished all BIFs equally. German wasn't satisfied. He wanted to know exactly who the diners were and what dishes they were ordering. To do that, he took a cue from history and assembled a diverse team of chemists, microbiologists, and food scientists. Together, they identified all the HMOs, pulled them out of milk, and fed them to bacteria. And to their chagrin, nothing grew. The problem soon became clear. HMOs are not an all-purpose food for biffs. In 2006, the team found that the sugars selectively nourish one particular subspecies called Bifidobacterium longum infantis, or B. infantis for short. As long as you provide it with HMOs, it will outcompete any other gut bacterium. A closely related subspecies, B. longum, grows weakly on the same sugars. The ironically named B. lactis, a common fixture of probiotic yogurts, doesn't grow at all. Another probiotic mainstay, B. bifidum, does slightly better, but is a fussy, messy eater. It breaks down a few HMOs and takes in just the pieces it likes. By contrast, B. infantis devours every last crumb with a cluster of 30 genes, a comprehensive cutlery set for eating HMOs. No other biff has this genetic cluster. It is unique to B. infantis. Human milk has evolved to nourish this microbe, and it, in turn, has evolved into a consummate HMOivore. Unsurprisingly, it is often the dominant microbe in the guts of breastfed infants. It earns its keep. As it digests HMOs, B. infantis releases short-chain fatty acids that feed an infant's gut cells. So while mothers nourish this microbe, the microbe in turn nourishes the baby. Through direct contact, B. infantis also encourages gut cells to make adhesive proteins that seal the gaps between them and the anti-inflammatory molecules that calibrate the immune system. These changes only happen when B. infantis grows on HMOs. If it gets lactose instead, it survives, but doesn't engage in any repartee with the baby's cells. It unlocks its full potential only when it feeds on breast milk. Likewise, for a child to reap the full benefits that milk can provide, B. infantis must be present. For that reason, David Mills, a microbiologist who works with German, actually sees B. infantis as part of milk, albeit a part that is not made in the breast. Human breast milk stands out among that of other mammals. It has 5 times as many types of HMOs as cow's milk, and several hundred times the quantity. Even chimp milk is impoverished compared to ours. No one knows why this difference exists, but Mills offers a couple of good guesses. One involves our brains, which are famously large for a primate of our size, and which grow incredibly quickly in our first year of life. This fast growth partly depends on a nutrient called sialic acid, which also happens to be one of the chemicals the B. infantis releases while it eats HMOs. It is possible that by keeping this bacterium well fed, mothers can raise brainier babies. This might explain why, among monkeys and apes, social species have more milk oligosaccharides than solitary ones, and a greater range of them to boot. Larger groups mean more social ties to remember, more friendships to manage, and more rivals to manipulate. Many scientists believe that these demands drove the evolution of primate intelligence. Perhaps they also fueled the diversity of HMOs. An alternative idea involves diseases. Pathogens can easily bounce from one host to another, so group-living animals need ways of protecting themselves against rampant infections. HMOs provide one such defense. When pathogens infect our guts, they almost always begin by latching onto glycans, sugar molecules, on the surface of our intestinal cells. But HMOs bear a striking resemblance to these intestinal glycans, so pathogens sometimes stick to them instead. They act as decoys to draw fire away from a baby's own cells. They can block a roll call of gut villains, including Salmonella, Listeria, Vibrio chlorella, the culprit behind chlorella, Campylobacter jejuni, the most common cause of bacterial diarrhea, Entamoeba histolytica, a voracious amoeba that causes dysentery and kills 100,000 people every year, and many virulent strains of E. coli. They may even be able to obstruct HIV, which might explain why most infants who suckle from infected mothers don't get infected, despite drinking virus-loaded milk for months. Every time scientists have pitted a pathogen against cultured cells in the presence of HMOs, the cells have come out smiling. This helps to explain both why breastfed babies have fewer gut infections than bottle-fed ones and why there are so many HMOs. It makes sense that they would need to be diverse enough to handle a range of pathogens, from viruses to bacteria, says Mills. I think it's the amazing diversity that provides a constellation of protections. The team is just getting started. They have set up an impressive milk processing facility in their mock Tuscan Institute to discover many unfamiliar secrets with this most familiar of fluids. In the main lab, which Mills runs with food scientist Daniela Barilli, there are two huge steel drums in which milk is stored, and a pasteurizer that looks like a cappuccino machine, and a riot of other equipment for filtering the liquid and breaking down into its components. Hundreds of empty white buckets are stacked on a nearby rack. They're normally full, Barilli tells me. The full buckets are kept in a huge walk-in freezer that's chilled to an intensely uncomfortable negative 32 degrees Celsius. On a nearby bench, there's a row of wellies. When we process, there's milk all over, says Barilli. A hammer for chipping ice, the door's not closing properly, and inexplicably, a ham slicer. I didn't ask. We pop our heads inside. White buckets are arrayed on pallets and shelves containing some 600 gallons of milk between them. A lot of this is cow's milk donated by dairies, but a surprising amount came from human breasts. Lots of women pump milk and store it, and once their kid weans, they think, now what do I do with it? People then hear about us and we get donations, says Mills. We got 80 liters collectively over 2 years from someone random at Stanford University who said, I have all this milk. Do you guys want it? Yes, they did. They need all the milk that they can get. Their plan is to study the components of milk, HMOs and beyond. There are fats and proteins with glycans stuck to them too. How do these affect B. infantis over other BIFs? Any other BIFs? There are phages as well, lots of them. German has teamed up with Jerry Barr to see if mothers use breast milk to provide babies with a starter pack of symbiotic viruses. They have already found something weird. Phages are great at sticking to mucus, but they do so 10 times more efficiently if there's breast milk around. Something in the milk helps them anchor in place. The culprits seem to be little spheres of fat encased in proteins that resemble those in mucus. If you let a glass of milk sit in the open, the layer of fat that forms on the surface is full of these globules. They provide nutrition to a baby, but they also might give baby's first viruses a foothold in the gut. When Barr tells me about this, I'm astounded. It means that the measures by which we shape and control our microbiome, the phages, the mucus, the various arms of the immune system, and the ingredients in milk are all connected. I've discussed them if they were separate tools, but they are all part of a huge interwoven system for stabilizing our relationship with microbes. In this counterintuitive reality, viruses can be allies, immune systems can support microbes, and a breastfeeding mother isn't just feeding a baby, but also setting up an entire world. And breast milk? German was right. It's far more than a bag of chemicals. It nourishes baby and bacteria, infant and infantis alike. It's a preliminary immune system that thwarts more malevolent microbes. It is the means by which a mother ensures that her children have the right companions from their first days of life, and it prepares the baby for life ahead. And there's one more note, this is note 39, that I also wanted to share because this is kind of fascinating to me, especially as a mom that definitely used breast milk as something of as a way of encouraging my little ones to settle down. We use that for sleep. We use that for boo-boos. We use that for all kinds of things. Let me see if I can find it. And here is note 39 from chapter 4. It could also be a way for mothers to manipulate their babies. It's in a baby's interest to monopolize as much of a mother's attention as possible, and evolution has given babies many ways of doing so: crying, nuzzling, and sheer adorableness. but a mother has to divide her care between many children, present and future. If she spends too much effort on any one, she might not have enough energy to raise more. So evolution ought to equip mothers with countermeasures. And evolutionary biologist Katie Hind suspects that milk is one of them. It nourishes specific microbes and, as we saw last chapter, some microbes can affect the behavior of their host. By altering the HMO content of her breast milk, perhaps a mother can, inadvertently, elect for mind-manipulating microbes that influence her babies in ways that benefit her. For example, if the infant is less anxious, it might become independent sooner, leaving mom to focus on her other kids. So, all in all, this was absolutely startling and stunning information. And this has confirmed everything that I sort of intuitively felt about breast milk and making the choice to breastfeed. But here you have it from a scientific perspective. Thank you to Ed Wong. And I really encourage you to go check out everything that he's done in YouTube. He's just a really fascinating author, and I love reading his work. It really is. He's also done some shorter articles that are cited throughout this particular work, and it has been just a delight to read his works. I wish that I could be doing that same kind of research. It just makes me excited every time I sit down to see what's going on, because really the relationship between us and our microbes is totally fascinating. But this bit as a mom makes me especially fascinated. And even though my kids are much older now, they're 11 and 6, and we're finally beyond the breastfeeding years. So yeah, it does end eventually, even though sometimes it might not feel that way. It's just really supported everything that I felt deeply about that. And, it doesn't always work out for everybody. I've gone to enough breastfeeding meetings to see that there are many people who really struggle with it for a lot of reasons. So if you can't do it, you can't. And that is something that happens sometimes. But if you can, this is an absolutely startling microbiological wonder. Really, a microbiological wonder that we as mothers are uniquely able to wield. Have a great one.